Hello everybody and welcome to Photosynthesis 2030, the webinar series aimed at promoting collaboration across the photosynthesis community. The webinars here are organised by Capitalize, an EU Horizon 2020 project that uses genotyping and phenotyping techniques to exploit natural variation in key photosynthetic traits for crop improvement. I'm Dr. Elizabete, and today I'm delighted to welcome my two guest speakers, Professor Elizabete Carmo Silva from Lancaster University and Professor John Cushman from the University of Nevada. Throughout the talks, we'd like them to be interactive, so we would like you to put your questions in the questions box during the talk, and then at the end of the presentation, I'll try and put these to the speakers. Our first speaker, Elizabete, is well known for her work on the regulation of Rubisco by Rubisco activase. In the Capitalize project, she leads the team at Lancaster, understanding natural variation in the photosynthetic efficiency and regulation in response to the environment. Today, she's going to talk about Rubisco isoform diversity and crop adaptation to dynamic environment. I'll hand that over to you, Elizabete.
Hello, everyone. It's a great pleasure to be here today and to be attending the Photosynthesis 2030 Plus webinar series. And today I will be telling you about the work my lab has been doing on Rubisco activase isoform diversity and how we can use this diversity to help improve crop adaptation to dynamic environments. And I'll start by showing you the team at Lancaster. So this is the photosynthesis team at the Lancaster Environment Center. We have 4 PIs. So in addition to myself, there's Professor Steve Long, Dr. Margarine Landgren, and Professor Martin Perry. And also on the screen, you see researchers and PhD students, master students, and technicians that work directly with me in this team. Our research aims to improve photosynthetic efficiency, and our key crops of interest are wheat and cowpea. And this research is funded through, in the case of wheat, through the BBSRC, and in the case of cowpea, via the RIPE project, which is funded by the Foundation for Food and Agricultural Research, the Foreign Commonwealth and Development Office, and the Bill and Melinda Gates Foundation. We also get some funding from the N8 Agri-Food. And so this work really aims to contribute towards a more sustainable agricultural crop production by making plants more efficient, produce more of our products with less resource. And when we aim to improve photosynthesis, I like to think of the photosynthetic process as basically a factory with many substations, which are the many sub-processes of photosynthesis. If we want to improve the efficiency of this process or the efficiency of our factory, we need to think about the various sub-processes and identify which ones are most limiting to our target crop in the environment where we want to grow it. In the case of wheat and cowpea, we know that Rubisco and its regulation limit photosynthetic productivity in these crops, so and for their target environments. So it is we know that there is scope for improving photosynthetic efficiency by tackling Rubisco and making its activation more efficient. Now, when we think about Rubisco regulation, Rubisco has this propensity to become inhibited. So what happens is that sugar phosphate derivatives bind to the catalytic site, and this locks the catalytic site in a non-productive conformation. At this stage, the sugar phosphate derivatives that act as inhibitors are not able to release themselves from the active site, from the catalytic site. And so Rubisco depends on interaction with Rubisco activase, which uses the energy from ATP hydrolysis in order to open up the catalytic site of Rubisco and enable the release of those inhibitors. And this renders Rubisco catalytic sites available for carbamylation and for catalyzing its activity, so carboxylation or oxygenation of our UBP. Importantly, because Rubisco activase is an ATPase and uses energy from ATP hydrolysis, is regulated by the redox conditions of the chloroplast, as well as ATP and ADP availability. Rubisco activase modulates the activity of Rubisco in relation to the amount of light that is available at the leaf level. Also, Rubisco activase is a thermal sensitive protein, and this means that at moderately high temperatures that are just slightly above the temperature optimum for plant growth, Rubisco activase in many species limits photosynthetic potential in those species. So it is an important enzyme in terms of regulating photosynthetic activity in response to the light availability at the leaf level, as well as in response to high temperatures. Now, when we look at the temperature response of Rubisco activase activity, this is reflected by a decline when the temperatures reach these slightly warm temperatures. So what is shown here is the activity of Rubisco activase in terms of catalytic sites of Rubisco that are activated per minute, and this was measured at different temperatures. And when we measured cool adapted species, the temperature optimum is considerably lower than the temperature optimum of warm adapted species. Now, if we think about the climatic changes that are happening and the increased frequency of heat waves and fluctuation in the temperature that plants are exposed to, we really want to see a temperature response that is more in line with this yellow line that you see here. So it's a broader temperature response so that we can maintain Rubisco activity at the cooler temperatures, but also at the warmer temperatures. Now, in wheat in particular, we know that, and it's been shown for a couple of decades, that Rubisco activation by Rubisco activase is inhibited at high temperatures, and importantly, this inhibition leads to decreases in crop productivity. So if we were to really broaden this temperature optimum, there is significant scope for making plants of wheat more productive at a wide range of temperatures. Now, in order to understand Rubisco activase diversity, what we started by doing, and this was work led by Joanna Scales when she was a PhD student in the lab, was to characterize what genes are present and what proteins. So in wheat, we have two genes of Rubisco activase, and one of these genes is alternatively spliced. This means that the expression of these two genes results in three different proteins. So gene 1 results in the formation of one protein that we call 1 beta, and beta 4, the short form of Rubisco activase. Whereas gene 2, by alternative splicing, produces both this shorter form, which we call 2 beta, as well as a longer form, which we call 2 alpha. Now, the longer form, you'll see, has a C-terminal extension with 2 cysteine residues that confer redox sensitivity to Rubisco activase. Now, what Joanna also saw was that by immunoblotting, was that the longer isoform here is present in considerably lower abundance compared to the shorter isoforms. And she couldn't distinguish the two short isoforms because they're approximately the same size, so we can't distinguish them on SDS page. But we knew that this isoform is present in lower abundance in the leaves of wheat. Now, Alejandro Perdomo, a postdoc in the lab, was interested in understanding the properties of these Rubisco activase isoforms. So we have 3 protein isoforms for Rubisco activase in the leaves of wheat. If we want to characterize their properties independently, we can't just extract them from the leaf, because we will have a mixture of all of those three isoforms. So he expressed each of the isoforms independently in E. coli and then purified them, and so looked at their properties independently in vitro. And what they did was to use this assay that looks at Rubisco activity, starting off with inhibited Rubisco. So we basically put an inhibitor in the catalytic site of Rubisco and then see what happens throughout the assay. What you're seeing here is the control reaction, where we have no ATP and no Rubisco activase, and here we see no increase in activity or very little increase in activity. So Rubisco activity remains inhibited throughout the 3 minutes of the assay. By comparison, when we add each of the Rubisco activase isoforms from wheat, Rubisco activity increases rapidly as Rubisco activase is removing those inhibitors from catalytic sites. And importantly, what Alejandro saw was that the two forms that result from expression of gene 2, so isoforms 2 beta and 2 alpha, were considerably faster at activating Rubisco compared to this other isoform 1 beta. So the catalytic rate of Rubisco activase isoform 1 beta is considerably slower than the other two isoforms. Now, we were interested in the temperature response, and Gustav Degen was a PhD student in the lab who was interested in characterizing this temperature response of Rubisco activase isoforms. So what is done was to measure ATP hydrolysis activity as well as Rubisco activation activity. So I mentioned before that Rubisco activase uses the energy from ATP hydrolysis to activate Rubisco. So it's actually doing two activities. It's hydrolyzing ATP and then using that energy to activate Rubisco. So he measured the two activities separately at different temperatures for the three isoforms. And there's a number of things to be seen here. So one of them is that one of the isoforms is slower at hydrolyzing ATP, but has fast rates of Rubisco reactivation, whereas the 1 beta isoform, for example, has high rates of ATP hydrolysis, but relatively slower rates of Rubisco reactivation. So this in some way means that the Rubisco activase isoform 1 beta is less efficient because it is using a lot more ATP at, say, a temperature of 30 degrees compared to a lower level of Rubisco reactivation. Now, we could also see a shift in the temperature optimum of these isoforms. And to better visualize this, Gustav expressed the data relative to the maximum activity for each of the proteins to really identify what was that temperature optimum. And so in terms of ATP hydrolysis, there was a shift towards higher temperatures by about 5 degrees, and the same was true for Rubisco activation. Now, another aspect was that in general, the temperature optimum of ATP hydrolysis, or the temperature at which the maximum rates were observed, tended to be higher for ATP hydrolysis compared to Rubisco activation. So, but the key thing from these results is that this isoform 1 beta has a temperature optimum that is about 5 degrees higher for both activities compared to the other two isoforms. Now, Gustav was interested to know whether there was a genetic factor affecting these differences in the isoforms. And so he looked through the sequences and identified a couple of amino acids that are different in the sequences of the isoform 1 beta that had the higher temperature optimum compared to the isoform 2 beta that had the lower temperature optimum. And there was one of the residues in particular that is an isoleucine in the more thermal tolerant Rubisco activase isoform, and the methionine in the other one. It's also an isoleucine in wild rice and cultivated rice species, which are adapted to warmer temperatures. So Gustav hypothesized that if he was to change this methionine in the isoform that has a lower temperature optimum to an isoleucine, this would increase the temperature optimum. He also produced the double mutant here, basically to verify that change in temperature optimum would be due to this specific change in the single mutant. And so he then went on to characterize the temperature response of these mutant proteins, where he had done the site-directed mutagenesis, and basically saw that in terms of ATP hydrolysis, there was a shift towards higher temperatures, more similar to the thermal tolerant Rubisco activase isoform, by changing that one amino acid. So changing a methionine for an isoleucine made these mutant proteins more similar to the thermal tolerant protein. In terms of Rubisco activation, what happened was actually that the mutant proteins, which are here in the light blue and turquoise colors, were more active than the isoform that had the lower temperature optimum. But they also maintained relatively high activity at the cooler temperatures. So this was rather exciting because basically it means that there's a broadening of the temperature response of Rubisco activase in these two mutant proteins. So this single amino acid change extends the temperature optimum of Rubisco activation. At about the same time, another group published some results showing that several mutations on the same protein, the one that has the lower temperature optimum, also increased thermal tolerance. And these mutations included the mutation that we were looking at. Now, Gustav was interested to know what happens in plants. So when plants experience heat stress, does the abundance of this particular protein increase? And so he grew plants of wheat at control conditions, and then at the booting stage exposed them to a heat wave. And this is relevant because it's the growth stage at which plants in the UK are at in spring, and we are seeing an increased rate or frequency of heat waves in the UK in the spring. The first thing to test was that the leaf temperature was indeed higher in the plants that were exposed to the high temperature. And this is because plants tend to cool their leaves when they are well watered, as was the case. And the temperature was considerably lower than the air temperature in the leaf there, but it was higher than under control conditions. So the plants were effectively experiencing a higher temperature at the leaf level. He also returned the plants to control conditions after the heat wave and then monitored their development through maturity and measured grain yield, and saw that having experienced that heat wave at this vegetative growth stage led to a decrease in the grain yield that plants attained at the end. So the heat stress was effective. What happened to Rubisco activase expression? Well, basically the 1-beta isoform, which had the higher temperature optimum, increased in expression relative to the other isoforms and responded relatively fast. So after 1 day at 38 degrees, it was already increased. And when the plants were returned to 25 degrees, the expression went down. Now, gene expression gives us a good indication, but we wanted to know what happens in terms of protein. So Gustav used the polyclonal antibody to determine the abundance of the alpha and the beta isoforms. But as I mentioned, we can't distinguish the beta isoforms with this antibody. So we developed a specific antibody for the isoform 1-beta that is more thermal tolerant. This isoform is present in very low abundance, almost undetectable under control conditions, and increases in abundance under heat conditions. So translating this information into numbers, the Rubisco activase content at the protein level increased in the heat-stressed plants, but only after, in the measurements done at 5 days of heat stress, and this abundance was still high when the plants returned to control conditions. The same was not true for the other isoforms, which remained constant throughout the treatment. Now, importantly, if you look at the numbers, the scale here is for lower numbers compared to the numbers here. So, although the abundance of the thermal tolerant isoform is increased, is still much lower than the abundance of the other two isoforms, so up to a maximum of 10%. So to summarize what we know about the diversity of Rubisco activase isoforms in wheat, we know that one of the isoforms has faster rates of Rubisco activation, and that is a more efficient enzyme, version of that enzyme. We also know that the isoform that is least abundant and least efficient nonetheless is interesting from the perspective of having a higher temperature optimum, and it increases in abundance under heat stress, but not to levels that are likely to cause much change in actual photosynthesis. We also know that one single amino acid change leads to a broader temperature response, and I didn't show those results, but this protein also showed intermediate efficiency between these two native proteins. So this really shows that there's scope for exploring diversity in Rubisco activase isoforms so that we can broaden the temperature response of photosynthesis in wheat and make plants more adapted to global warming and the fluctuations that brings along. So in my final research slide is just to show this figure where I highlighted here what are the isoforms that are present in model species Arabidopsis and tobacco, and what's their protein length light response, which I didn't go into detail here today, but also in terms of temperature response and isoform abundance, and what we know now for the wheat isoforms. So we know that they differ in length. We know that two of them are considerably slower than the other one in terms of its light response. And we know that another one has a higher thermal tolerance. And we also know what is their usual abundance in wheat leaves growing under control conditions and in the field. So this started to really build a story that we can now explore in terms of trying to identify natural variation in wheat germplasm, as well as create novel variation by gene editing technologies in order to explore single amino acid changes that can lead to significant changes in the temperature response of Rubisco activase. So with that, I'll finish by thanking the team again, and I mentioned their names of the people that were most involved with the work as we went along, and I also thank you for your attention.
Thank you, Elizabete. That was a great talk. I've got one burning question that I'm desperate to ask. Actually, I mean, I presume you get a range of you would expect a range of isoforms in most plant species. Is that right?
Yes, so there's some species where you have only two isoforms, and there's some species where you have quite a lot more diversity of isoforms, yes.
So and I know looking through some of the Rubisco activase sort of papers, a lot of people talk about C4 plants where they look for variation, but surely one of the places, and maybe John might be able to back me on this, that you might find really big differences in the cam plants that are, growing normally at 40 degrees or whatever. Would you not expect them to have huge amounts, the opposite way around, of the beta protein or the beta isoform?
Yeah, so, and there's some research done on cam plants, in particular agave, for example, and they did identify some interesting properties of Rubisco activase. But as far as I know, that research group that has been studying cam Rubisco activase hasn't yet published what is the diversity that is present in those plants. So there is information about the Rubisco activase from those plants, but not in terms of its diversity. So I think it is a very exciting field of research. John, I don't know if you have something to add to that. Quite curious.
Yeah. Well, I think if I'm remembering correctly, the activases were more active or stable at higher temperatures. So there is definitely some prospects there for higher temperature tolerance.
Yeah. I have a question here from Chris Rains. How common is it to have only one isoform?
Right, so in the species that I'm aware of, there's typically at least 2 isoforms. Whether they are longer and shorter isoforms or just short isoforms, then that's another question. So, for example, in Solanaceae species, the longer version of Rubisco activase is not present. So the C-terminal extension has been lost through mutations and during evolution. So in Solanaceae, there's only the shorter version of Rubisco activase, but nonetheless, there's more than one gene. So whether then those short isoforms differ in their properties or not, I think we haven't yet looked into that.
From Tracy Lawson, how often through the day does Rubisco activase have to remove sugar phosphates from Rubisco? And what determines when sugar phosphates bind to Rubisco?
Wow, that's a really good question, Tracy. So basically, this is happening constantly because there's always a fraction of catalytic sites of Rubisco that will be inhibited. So this is required throughout the day, and it's just the proportion of it that changes. So at higher light, there's less inhibitors that are binding. Compared to lower light, and this is determined, for example, by the magnesium concentration in the chloroplast, as well as the pH and the carbamylation level of Rubisco, which I didn't get into that detail here today. And the other thing is the temperature. So at higher temperature, there's more inhibitors. So those fluctuations in light and temperature will determine what extent of Rubisco is active or inactive, and therefore how much Rubisco activase we need at each time.
It says, hi, it's from Jonathan Menery. Hi, I'm a social scientist with a project called PhotoBoost, also involving Lancaster. We are working with various stakeholder groups, and we often get asked by the more GM skeptical types, why do we need to improve photosynthesis? It would be great to have your response to this. So, where do you start?
Yeah. Elizabete, you want to go first?
Sure. I guess why do we need to improve photosynthesis? Well, if we want to improve the genetic yield potential, so the productivity of plants, without increasing the amount of resources that we're using, so in a way to improve the sustainability of agriculture, this is a great way to do it, because we can really do more with less, or at least with the same amount that we have currently. So I guess it's recognizing that we have finite resources in the world, and improving photosynthesis is one possible way to produce more with less. So that would be my response.
Okay, thank you for your very comprehensive answers. And it just remains for me to thank both our speakers now, because I think we need to wrap it up, but 2 really interesting talks, and to the listeners for some great discussion. I'd like to remind you that the webinars take place on the first Tuesday of each month, and that our next webinar will be on the 7th of December with Professor Roberta Crochet and Dr. Marjorie Lundgren. So Please join us for those. Don't forget you have to register. And again, once more, thank you so much for a really interesting webinar this month. Goodbye, everybody.
Thank you.
Thanks, everyone.
Bye-bye.
Bye.
Bye-bye.